Hello everyone. In this video I'm going to explain Respirometer. What is Respirometer? Respirometer is a device in order to measure the amount of the respiration. This is a very simple and very informative picture that I'm going to show you how a respirometer works. Simply, we have two bottles, and in one bottle we have an insect. This could be an insect, or this could be a seed germinating, any organism that have the activity or live activity that does the respiration can be used for these type of experiments. Actually we cannot do this experiment in our IB because we cannot use insects in our experiments. It's against the IB policy. However, for your understanding, I just explain from this experiment, and we will do like a virtual lab using the insect that is really virtual and nobody is going to kill anyone. So that would be very nice and okay even with the IB. So let's go back to the experiment. How should be the setup and how does it work? In this bottle we have the insect, and as you can see there is a carbon dioxide absorbent. Well, what happens here, we are imagining that we have oxygen molecules, right? So we have oxygen molecules over here, and also if the insect is doing the respiration, we also have the carbon dioxide molecules due to respiration, because it absorbs oxygen and it releases the carbon dioxide. Yeah. The purpose for this carbon dioxide absorbent is that it absorbs the carbon dioxide due to the process of the respiration. And what happens here, the only thing which will be remained in order to be used for the insect is oxygen. Once the oxygen will be absorbed by the insect, already carbon dioxide goes here. So the only thing that shows the usage of the oxygen can be monitored by the movement of this colored oil. And pause the video and tell me what happens if the oxygen here is used by the insect. Carbon dioxide will be absorbed here, oxygen will be used by the insect, and what happens to this oil color? I give you just some tip. This oil color will move. What do you think? Is this movement would be like this, or the movement would be like this? Pause the video and answer. Okay, I think you thought, and maybe you answered correctly. I hope you did. The answer is the colored oil will move up. So this is not correct. Why? Because the oxygen will be used and there is no carbon dioxide, because it will be absorbed by this absorbent, and the volume will decrease over here. And once the volume is decreased and the pressure, sorry, not the volume, the pressure is changed, and once the pressure, because of the usage of the oxygen by the insect, will be changed and decreased, the low pressure over here is sucking, you can say is sucking the oil, or it's the better word is absorbing, or putting a pressure on the colored oil to move up. So the colored oil will move up, like goes like until here, and this goes down. So we will have the change of the colored oil from here and move up over to here, because of the oxygen has been used over here. Okay, second question. What do you think is this bottle for? What is the role of this bottle? Pause the video. Okay, this bottle is for controlling. Why? Because we need to have the same amount of the absorbent, and we need to know that, for example, if in this bottle, if there is oxygen or carbon dioxide, there will be equally absorbed by the absorbent. So whatever is causing the change or the movement of the oil color should be just the usage of the oxygen. That here, because of the insect, the oxygen will be used, but here we don't have any insect, so there is no one that will be used oxygen. So the level is not changed and nothing happened in the pressure at this bottle. So that's the way that we can control and say the only reason that the oil is moving is because of the usage of the oxygen. Okay, so that was the first part of the explanation. Let's go to a real experiment. I mean, the previous one also was real, but this one is something with more advanced parts and includes the seed, which is germinating. It's the same process. The reason that I explained the previous one because it was much more easier to catch up with what's happening in the real experiment. This is exactly the same thing. We have a seed germinating over here that is going to do the respiration, and this means that it absorbs the oxygen by doing the respiration and produce the carbon dioxide, but the carbon dioxide moves down over here because of the KOH, or the absorbent of the carbon dioxide. So we have the low pressure over here because of the absorbing the oxygen, and then the color, the oil color will move up. And we have the same tube here, but this is a control, and this control doesn't do the respiration. Exactly the same thing. So the only thing is happening here, and the only difference between this tube and this tube is that here the oxygen will be used, but here no. And that would be the reason that we can judge that this is the respiration is happening, because the oxygen only will be used. So let's see what are the boxes over here, one by one. This is hypodermic needle. That's a syringe. The syringe is for setting up the experiment and make sure that the level and everything is has been added properly, the amount and whatever it has been used for adding the oxygen, for example, here, so that we can know that how much and how the experiment can be set up equally for both. And then respirometer tube, both of them. We have water bath. Okay, this is something that I didn't explain over the previous slide. What do you think water bath is for, and what is the role of the water bath? Pause the video. Okay, water bath is for keeping the temperature constantly between these two, and also to make sure that because the temperature will affect the volume of the gas, and it changes the volume because it's the gas, and it makes more movement of the bottom of the gas molecules, and in that case will affect the movement of the oil, and it affects the pressure. That's why we use the water bath, to keep it constantly, not to be affected by the temperature. So we set up a constant temperature. Yeah, so the respiring organism give up carbon dioxide and absorb oxygen. That's what I said. And here is the absorbent, and carbon dioxide is absorbed, and then at this stage, the manometer fluid moves up to the right because of the change in the pressure, and any change in pressure or temperature during the experiment affects the respirometer tube and the thermobarometer tube equally. So this shows the as a control and a constant between both and equal between both temperature and everything except the usage of the oxygen by the organism. Yeah. After a fixed time, a syringe is adjusted to the level. The fluid is in the two arms of the manometer, and the volume of oxygen absorbed is read off on the syringe. The amount of the oxygen that has been used up by the organism can be set up by here. Sorry, can be read by this syringe. So if the oxygen will be absorbed, this means that not only it reduces the pressure over here, so the movement of the color oil is here, but also the amount of the oxygen that has been used or has been absorbed can be seen from this syringe as well. How much oxygen has been taken up from the syringe to this tube in order to be used for the respiration. Okay, I think I already explained this in the first slide. Respiring organism, if using invertebrates rather than seeds, what are the ethical questions that need answering? So many things. Is it acceptable to remove animals from their natural habitat for use in an experiment? No, we cannot do that in IB, but you can do it in university when you go there, just not in IB. Can the animals be safely returned to their habitats? Yeah, that's what that you have to answer to that question. If you can use the insect or any animal that you can safely return without any harm, then you have to prove it, and then no problem with your IB experiment. Will the animals suffer pain or any harm? You cannot do this. You cannot confirm if the animal is suffering or not. So no way. You have to just give up from using the animal. Can the risk of accidents that cause pain or suffering to the animal be minimized during the experiment? In particular, can contact with the alkali be prevented? Is the use of animals in the experiment essential, or is there an alternative method that avoid using animals? I think this is the key question at the end. Once you have the alternative, there's no way that you want to use an animal. This is similar to like the enzyme experiment. If you can use potato, there's no need to use liver for doing that type of experiment. There is a link over here. It's the respiration lab, and you can do this virtual lab. But there is another one which I like. And I'm asking you to do this virtual lab. You go to this link, and it's because we have done this virtual lab before in the class. I mean, not this virtual lab, I mean general virtual lab. You know what to do. You just start to from the first page, and step by step go and do it. And it's in this virtual lab. It's the same is happening. Which I explained in my first slide. The oxygen will be absorbed by the insect from here, and then the KOH is absorbing the carbon dioxide. So the only way that will be measured is the usage of the oxygen, which shows the amount of the respiration.
Hi, this is Mr. W from ScienceMusicVideos.com, and this video is about measuring cellular respiration with a respirometer. During During cellular respiration, living things take a fuel like glucose and oxidize it to create ATP. The waste products are carbon dioxide and water. The rate of cellular respiration, how much of it happens over time, depends on the organism, its activity, and its environment. In a high school or college biology lab, with a small organism like a germinating seed, we can measure the rate of respiration with a device called a respirometer. Here's how it works. There's a glass chamber, like a test tube or a small bottle. At the opening, there's a 1 milliliter pipette that's been jammed through a rubber stopper. And when the rubber stopper is in place, the only opening into the system is through this hole. Our respiring organism goes in here, and then there are 2 layers of cotton. One is absorbent cotton. We're going to soak that with KOH, a strong base, and what that does is it chemically absorbs all the carbon dioxide that's produced by the respiring organism. And then there's a layer of non-absorbent cotton whose function is to protect our respiring organism from that strong base. You seal off the top of the pipette with food coloring, or you can immerse the entire system in water. Now there's a fixed volume of gas inside the respirometer. As your organism respires, the CO2 that it produces will be absorbed by the potassium hydroxide that's in the absorbent cotton. The organism itself will consume the oxygen molecules in the chamber as it performs cellular respiration. That will cause the gas volume in the chamber to decrease, creating suction that will draw the food coloring down the length of the pipette. And here's what we'd see using time-lapse photography if we observe the respirometer for about 20 minutes. But how do we know that it's the disappearance of oxygen that's drawing the food coloring down the length of this pipette? How do we know that it's not gravity or a change in the surrounding air pressure? We need a control. If our assumption is that a respiring organism is consuming the oxygen inside that chamber, then we need to see what would happen if there's no respiring organism. To do so, we build an identical respirometer, but instead of our respiring organism, we substitute an equal volume of something that's not respiring, like these plastic beads. To make sure that the volumes are equal, you can use a method like measuring volume by displacement of water. Now let's observe this again with a control. Notice that in the control respirometer, the level of the food coloring isn't changing. And that means that the gas volume inside the respirometer isn't changing. The only difference between these 2 respirometers is the presence of our respiring organism. Let's finish by talking about how you determine the rate of respiration. The entire volume of this pipette is 1 milliliter. So if as we're doing our observations, we see the food coloring move from here, which is 0.8, to here, which is 0.6, in 10 minutes, then that's a difference of 0.2 milliliters in 10 minutes. And you can reduce that to 0.02 milliliters per minute. That's our rate of oxygen consumption for this many lentils at room temperature. So now that you know how a respirometer works, you can do all kinds of inquiry. Think about different variables like different types of seeds, or how long it's been since they germinated, or using different kinds of organisms all together. In every case, you're going to think about how to test those variables, and you're going to have to design an experiment with adequate control.
Here is a drag and drop, and you will learn by doing this drag and drop where to put the molecules from right side to the left side, and how to find the differences between the anaerobics and also with aerobic. And this is a good comparison for learning or reviewing, because this is also something that you need to learn here, and it will be reviewed on the Unit 8 for HL as well. So pause the video and drag and drop. Okay, I suppose that you have done drag and drop. So you need to find step by step the molecule which is starting to get broken down, which will be glucose. So glucose should go over here, and then will be broken down. And what is going to break down the glucose? Then that would be the ATP, and you know that. Is there a small yield of ATP or high yield of ATP? So this is the first step, and it's happening in the cytoplasm, which is anaerobic. So that would be the small yield of ATP here, which will be produced by this first step, anaerobic respiration. And what happens here, and what is the product of the first step, is that the 2-pyruvate is produced in here, and then 2-pyruvate is going to go two ways, depends on if it is going to no oxygen. So the no oxygen is going here, because if this is anaerobic, and if there is oxygen, then oxygen is going here, through the aerobic respiration. And what happens over here then? What is the product of anaerobic respiration? That would be either ethanol or lactic acid. Who is producing ethanol? Of course, yeast is producing ethanol. And who is producing lactic acid? Of course, animals such as us, in the muscle contraction, they will produce lactic acid here, and here is the ethanol. So that's all. And this is sugar, I think. Okay. I can also say what happens here. This is the aerobic respiration, and the product of the aerobic respiration, finally, the final stage would be like carbon dioxide and water molecules, which go to the last step in the aerobic respiration. So let's see, everything is placed in the next slide. Yeah, I did it correctly, you know. Good job. Bingo. So there are something happening in the breath and anaerobic respiration, and every one of you have seen the bread which is made by water, by adding water to flour. Do you have any experience of making bread? I think you do. Or even not, you have seen that in the bakery, what happens. So the process is adding water to flour, and then after adding the water, then there is some other ingredients that you add to your water and flour mix, which that ingredient is creating a bubble gas, and that would be like yeast. Why it does so? Because immediately after you put all the mixture, water, flour, and then the ingredient of the yeast together, then you put it in a warm bath or in a warm oven. If you have seen in a bakery, they have the ovens, and the temperature is warm, like more than 40 or like 30 plus, I think like 41, 42 degrees. And then what happens in that situation, in that condition, the yeast start to respire aerobically, but because the oxygen will be used so fast, then there is no oxygen anymore, and yeast start to do the anaerobic respiration. And in both cases, at the start of the respiration, which does the aerobic, the product would be like carbon dioxide, and once the oxygen is used up, then it goes to anaerobic, and then it ends up with the production of the ethanol at the end as well. So the carbon dioxide inside the bread cannot escape, so that's why that carbon dioxide in the bread forms the bubbles, and it causes that the dough swell and rise, and that's what you see after putting like 30 minutes or 40 minutes inside the oven, then the dough starts to swell out, and that's because the release of the carbon dioxide inside. And then what happens to the ethanol? Because ethanol is produced by the anaerobic respiration, it has the evaporation, it will evaporate very fast, so that's why you don't feel the ethanol inside the bread, because it's already evaporated. This explains the bioethanol, which is produced by the ethanol, which is produced by the organisms. How? So the process is that the bioethanol is produced from the sugar cane and maize, or the product which can be used for this bioethanol by the usage of the yeast. How does that work? The starch and the cellulose in the plant material, they are broken down by the enzymes into sugar. So the key point is sugar, and once the starch and cellulose from the sugarcane and maize are broken down using the enzymes to the sugar, then these sugars are exposed with the yeast, and the yeast is already in the optimum condition and start to do the anaerobic respiration. Once the yeast is doing anaerobic respiration, the sugar in plant will be converted into ethanol, and then that ethanol actually will be purified by distillation, and then water will be removed. And actually, this is not just a this is a bioethanol for industrial usage. However, you know that the alcohol industry also is using the same thing, and production of different types of alcohol would be the same, like wine production, beer, and so all of them are following the same protocol and the same chemical reactions. As we saw, anaerobic respiration, the first, the right part of that map that I showed you was related to the yeast anaerobic, and now we are talking about the anaerobic in animals, specifically in human. The human activities used anaerobic respiration because of the immediate requirement to the ATP. And like the weightlifting and sprinting is one of those things that you are using your muscle very fast and immediate, and that the oxygen is not completely enough for those muscle cells. So they start to do the anaerobic respiration, and for the supply of the ATP very rapidly they do the anaerobic respiration. And what happens is that the result or the product of the anaerobic respiration in muscle cells are lactate, and the lactate cannot be concentrated, or your body cannot tolerate the concentration of lactate more than a certain amount. So that's why also you feel pain in your muscles. And what happens is that that lactic acid must be broken down, and it happens very rapidly once the oxygen is provided. So as you can see, this involves the use of oxygen, and it can take several minutes for enough oxygen to be absorbed for all lactate to be broken down. After a couple of minutes, then that lactate can be broken down if the oxygen is provided. And the demand for oxygen that builds up during a period of anaerobic respiration is called the oxygen debt. That's all for this slide, and hope you enjoyed it. Thank you for watching.